Hi, my name is Tom Magliery. I'm the co-founder and CEO of Cogen Bioscience. Cogen is dedicated to developing innovative cancer targeting solutions for new therapeutics and companion diagnostics. It's probably happened to someone you know, a loved one, a friend, maybe even yourself. Cancer diagnoses are some of the most frightening. But what we want our physicians to have the best tools available to be able to eradicate the cancer and keep the patient safe. There have been remarkable advances in cancer treatments in the last few years, including immunotherapy and new targeted agents against leukemias and lymphomas. But it's not that widely appreciated that the treatment of solid tumors is far behind that. The best imaging tools for most kinds of solid tumors are CT, basically the same technology of X-ray. And most of the agents that are used against solid tumors are chemotherapeutics, which are toxic to cancer cells, but also the regular cells in our body. Why are we in this situation with solid tumors? Well, there's really two reasons. One is the molecules that we use to target cancer, and the other is the targets themselves. For a long time, the best molecules we've had available are full-length antibodies, and many of these molecules have elicited immune responses, have been difficult to produce, have been difficult to store or result in aggregation, or result in limiting toxicity because they themselves have functions. The other reason is that it's been very hard to find targets that are highly specific to solid tumors, that attack the solid tumor, but don't also attack natural cells. This is the advantage that Cogen has. Not only have we engineered a set of molecules that solve the problems with full-length antibodies, but also we're developing molecules against a target that is found in adenocarcinomas, over 70% of solid tumors. Nearly 1 million people a year in the United States alone develop new adenocarcinomas. The first piece that Cogen has is a breakthrough platform for targeting cancer. We've engineered out just a fragment of the antibody, made it extremely stable, made it essentially invisible to the immune system. And then we've gone one step further, and we've engineered it so that we can dial in the amount of time that these molecules spend in blood. Long amounts of time for therapeutic applications, shorter amounts of time for same-day imaging applications, and very small molecules that we can use for laboratory diagnostics. This gives us a panel of molecules that all hit exactly the same target, are extremely well behaved, and can be used together to treat patients highly effectively. The second advantage we have is a marker called TAG72, which is found in very high rates on the surface of many kinds of adenocarcinomas, including colorectal cancers, ovarian cancers, and pancreatic cancers, for which there are no approved targeted solutions. This means that Cogen's molecules will be able to change the treatment of a huge number of patients in the United States and around the world. The other thing about TAG72 as a marker is that it's already been in many patients. Actually, the first imaging antibody that was approved, Oncocyte, targeted TAG72. And it was used for over a decade in the clinic. But it was eventually withdrawn because it had some of those problems that I was telling you about. You had to inject it a week before it could be useful for imaging, and that just wasn't compatible with the process of care. But importantly, it found metastatic tumors, especially lymph node involvement that's so common with adenocarcinomas. Another company was developing a second generation molecule against TAG72, and they showed that if you removed all TAG72 positive tissue in surgery, that twice as many patients survived to 10 years. That's a tremendous therapeutic effect. But the molecule that they were using was difficult to produce and ultimately had to be withdrawn from studies because of production issues. Our molecules have solved that problem. They're stable, they're easy to work with, they're easy to produce. And we've done extensive imaging in live animals using colon cancer cell lines implanted into mice, where we see contrast as high as 30-to-1, which is industry leading. Also, we've done extensive laboratory immunohistochemistry that shows that these molecules are hitting the right target. And not only that, do better than the molecules that are currently in the lab at identifying certain kinds of adenocarcinomas, like breast cancers. Cogen's business plan is to develop a suite of targeted therapeutic molecules against adenocarcinomas, and to have companion diagnostic molecules for imaging, staging, and laboratory diagnosis. Our main goal initially will be to develop both alpha and beta emitter radiopharmaceuticals against antibody drug conjugates as our lead therapeutic molecule. Cogen has licensed the entire portfolio of these molecules from Ohio State University, where they were developed in my lab in the Department of Chemistry and Biochemistry. And not only that, we have the worldwide rights to the parent molecule that binds to TAG72 for greater freedom to operate and protection. In the last several years, there's been a renaissance in the use of radiopharmaceuticals. The first round of radiopharmaceuticals largely were withdrawn from the market because, frankly, the molecules that we were using, the antibodies, we didn't understand them well enough. But now we're entering a phase where we do have excellent targeting molecules. In the last 5 years, Novartis alone has spent $6 billion acquiring companies with radiopharmaceuticals for either therapeutic or imaging applications. Recently, the European company Bracco acquired Blue Earth Diagnostics for $500 million for an imaging solution against cancer. And a startup company, Fusion, recently has gone public and now has a market cap of $500 million. There was truly a tremendous amount of excitement in the medical community about the development of radiopharmaceuticals as new modalities that will both be able to treat and will be able to guide the treatment in a personalized medicine modality. Cogen is currently looking to raise seed money to complete animal studies on its initial therapeutic molecules to identify the lead molecule that it will take forward. At the same time, we'll do additional imaging studies to validate its use against additional cancers, and we'll carry out IND-enabling experiments that will pave the way to approval in a faster manner. I founded Cogen with Jonathan Savar, who is a former investment banker and a lawyer who specializes in mergers and acquisitions. And we've charted a path with our advisors for a rapid turnaround for our investors. We're being advised by Mike Tweedle, who's a research radiologist, and who also is the former president of Bracco Research, so he has extensive drug development experience. And also Ben Swanson is a clinical pathologist who specializes in this area of cancer. Cogen Bioscience is well positioned to develop innovative targeting solutions against adenocarcinomas that will change the conversation that doctors have with patients around solid tumors.
One of the questions that came through the chat asked, you mentioned that the molecules are easy to produce. Do you anticipate any challenges as you scale up production?
We really don't. The fragments that we've developed here are all E. coli expressible. And so the yields are actually quite good, and it's very easy to scale those up by volume. So you can do gram production from 200 liter type of production. And we've already done fermentation at much smaller scale to demonstrate what those yields would be. But we're talking about microgram doses in the imaging space and milligram doses in the therapeutic space. So we anticipate very low cost of goods sold.
Thank you. Tom, you mentioned that the treatment is for a few indications for which there aren't any good treatments currently. What indication do you plan to pursue for your first clinical trial?
Yeah, thanks, Cheryl. I think that the most obvious things that we can be pursuing are colorectal and ovarian because of the clinical history with TAG72 agents in that space, as well as pancreatic, because it's essentially an orphan type indication. So there's the potential for speeding that approval because there's really nothing in either the therapeutic and certainly targeted therapeutic or diagnostic space. So I think we envision the idea of going after a sort of all-comers study in colorectal and pancreatic as our starting place, and expanding that into additional trials.
Can you talk a little bit about the kind of price and margin you think is possible on the therapeutic and the diagnostic side for this product?
Sure, yeah. I think that, so as I said, the cost of goods sold for making these, especially since they're from really scalable production is really low. It's not essentially a main component of what you'd be spending. It'd be things like marketing that would really be the initial cost. And radiopharmaceuticals and especially some of the companion agents would need to go in with partners that really were specialized in those areas. But those exist. Those sales pathways already exist. If you look at agents, there aren't that many agents that are comparable right now. But for example, against neuroendocrine tumors, where there are both radiopharmaceutical, therapeutic, and imaging agents, the imaging agents, net spot, are reimbursed at about $3,000 to $4,000. And as I said, there's a very large market for this in the United States alone, approaching 1 million new adenocarcinoma cases a year. On the therapeutic side, it's much higher. For example, Lutathera, which is the companion